Did you know there's life beneath your feet that can extract gold from rock? Not with brute force, but with chemistry. Fungi, nature's quiet recyclers, have a hidden talent. They can mine gold. In this video, we'll explore the full journey. From the biology behind it, to how you could, in theory, use fungal biotechnology to extract gold from ore, waste, and even electronic scrap. Welcome to the underground science of gold bioleaching. With fungi, gold. It's captivated human civilization for millennia. Wars were fought over it, empires built with it, and economies founded upon it. But gold is hard to find. Traditionally, it's extracted through massive mining operations, involving cyanide leaching, heavy machinery, and environmental destruction. But now, the game is changing. Enter the fungi. Biomining, or biological mining, is an emerging field that uses microorganisms to extract metals from ores and waste. While bacteria like Acinetobacillus ferrooxidans are well known in copper and uranium mining, certain fungi have proven adept at scavenging and even precipitating gold. They're cheap, scalable, and far less damaging to ecosystems. Let's talk species. The star of our show is Fusarium oxysporum. This common soil fungus has an extraordinary ability. It can solubilize gold from its mineral matrix and precipitate it as nanoparticles. How does it work? It begins by secreting cyanide-free metabolites, such as oxalic acid and other organic acids, that can leach gold into a soluble form. Then, it produces reductive enzymes like NADPH-dependent reductases that convert dissolved gold ions into solid nanoparticles, often embedding them in a matrix of mycelium. Other species with potential include Aspergillus niger, strong acid producer, often used in bioremediation; Penicillium chrysogenum, capable of reducing gold ions; Phanerocate chrysosporium, a white rot fungus known for its lignin-degrading enzymes. But for gold mining, Fusarium oxysporum leads the way. Let's simplify the process. 1. Exposure to gold-containing material. You start with gold-bearing ore or e-waste, such as crushed circuit boards. The gold isn't visible, it's bound in rock or alloy. 2. Fungal cultivation grow Fusarium oxysporum in a nutrient broth, like potato dextrose broth, PDB, under sterile conditions. Incubate it for 3 to 5 days until you get a dense mat of mycelium. 3. Contact phase. Introduce the fungus to the gold-bearing substrate. This can be done in flasks or bioreactors. The fungus secretes organic acids, like oxalic acid, citric acid, and gluconic acid that dissolve gold into solution. 4. Reduction and precipitation. Once gold ions are in solution, the fungus's enzymes, like nitrate reductases, reduce them to elemental gold, forming visible gold nanoparticles. These collect in or on the mycelium and surrounding media. 5. Harvesting. After 3 to 7 days, filter the fungal biomass. Use centrifugation or drying to recover the gold nanoparticles. These can be smelted or sintered into larger masses if needed. To try this in a lab-scale setup, here's what you'll need. Fungal culture, Fusarium oxysporum, from a Mycology supplier; potato dextrose broth, PDB, or Sabouraud dextrose agar for initial culturing; gold-bearing ore or e-waste, preferably ground to fine particles; pH meter, shaker incubator, 25-30 years, 100-150 RPM; Erlenmeyer flasks, 250-500 ml well; centrifuge, optional but ideal; filters or filter paper; autoclave for sterilization; laminar flow hood, recommended for sterile culture. Safety first. While Fusarium oxysporum is not highly pathogenic, it can be an opportunistic pathogen. so always wear gloves, goggles, and a lab coat. Use a fume hood when handling fine dust or acidic solutions. Environmentally, fungal gold recovery is far superior to cyanidation, the traditional method that uses toxic cyanide. Fungal biomining uses renewable biological systems, produces minimal toxic waste, and has potential for circular recovery from e-waste. To scale this process, move from flasks to stirred tank bioreactors, 1 to 10 litres. Control pH, ideal W 5.5, and temperature, 25-30 palnisam. Use peristaltic pumps for feeding gold solution or ore slurries. Harvest the biomass in batch or continuous mode. Consider membrane filtration for gold nanoparticle recovery. Pilot studies show this is viable, especially in recovering gold from low concentration sources, such as tailings or scrap electronics. Nothing is perfect. Yield. Biomining is slower than chemical methods. Yield depends on particle size, gold concentration, and fungal strain. Selectivity. Other metals, copper, nickel, may interfere with gold binding or precipitation. Stability. Maintaining pure fungal cultures at scale is difficult without contamination. Regulatory hurdles. Commercial bioleaching for gold is not yet widely approved or standardized, but ongoing research is solving these issues with genetic engineering and process optimization. A 2020 study published in Nature Communications by Frank Raith et al. Revealed how fungi dissolve gold and precipitate it in nanoparticles. They isolated strains from gold-rich soils in Australia, showing that fungi play an active role in forming secondary gold deposits. Another 2024 paper, published in Frontiers in Microbiology, explored engineered fusarium species that increase yield by over 50% using enhanced reductus expression. In India and Brazil, pilot fungal bioreactors are being tested to recover gold from e-waste, showing strong potential for urban mining. This isn't just about gold. Fungi can extract palladium, platinum, silver, rare earth elements. Imagine a future where mycelium farms are placed next to landfills and mine tailings, harvesting precious metals in a green and quiet way. With CRISPR and synthetic biology, we could design super fungi tailored to specific metal targets. Bioleaching could become as common as composting. Gold mining using fungi isn't science fiction anymore. It's science in action. Slow, sustainable, and smart. While we're still years away from large-scale commercial fungal gold extraction, the groundwork is being laid right now. This method could revolutionize how we view waste, electronics, and the very soil beneath our feet. So next time you walk through a forest or see a patch of mold growing silently on a tree, remember, it might be more valuable than it looks. If you found this fascinating, share it. Comment below if you'd like a DIY video version of this process or a deep dive into e-waste mining with fungi. And don't forget to subscribe to Broad Science for more deep dives into the unexpected intersections of nature and technology. Until next time, think deep, think green, think fungal.